Okay, on to our next biomolecule. Now we're going to talk about proteins, which I'm sure you may have the most familiar with. So proteins all around our body, hair, fingernails, also muscle, tissue, going to be full of protein. If you work out and get bigger, a lot of things that people will consume will be a protein shake to assist with the repair of the tearing of the muscle tissue as you try to get bigger. But if you do too much too soon, that can lead to bigger injury. Transporting substances across the cell membrane. We'll talk about that probably in Unit 5 when we talk about how things get across the cell membrane. We know that the cell membrane is the lipid bilayer, and it's very good at keeping things out because it does have polar and non-polar components. So there are some ways around it. So we do have what's called membrane proteins. And then finally our enzymes, which your enzymes are known as your biological catalyst. And if you recall, what that means is they speed up reactions. Our elements, we still have C, H, and O, just as everything has had so far. We are now adding nitrogen. And then we also have sulfur. So the only thing we do not have in play with a protein is phosphorus. Everything else is in play. 5 of the 6. Now our building block, or our monomer of proteins, is the amino acid. And our body has specifically 20 different kinds of amino acids. Now when we look at an amino acid, we have the same basic form to an amino acid. We have what's called an amino group. We have just a hydrogen right here, and we have a carboxyl group. And finally, we have our side chain. This is going to be the difference maker in regards to which of our 20 amino acids this will be. So if we look down at the bottom here, notice how they both have the amino group. They both have the hydrogen. They both have the carboxyl group. But now, when we see what else is bonded, the R group is different. And since the R group is different, that's what leads to different identities of your amino acids. We have 20 different R groups, which leads to 20 different amino acids. Now, when we look at putting amino acids in a chain similar to your carbohydrates, instead of saying it bonds covalently when we make that bond, we call it a peptide bond. So a peptide bond is one that is specific to proteins. Amino acid bonding to amino acid. What we have here is the OH of one bonds to the H of another, so our carboxyl group of one bonds to the amino group of the other. When this occurs, H2O is lost. And what happens is we form a bond between the two. Does this ring a bell? What reaction is happening here? Whenever we are building, we have dehydration synthesis, where we lose water acids bonded together. We would call that similar wording to what we call with carbohydrates. Instead of disaccharide, which is two monosaccharides bonded together, we still have di, but now we simply take that word of what bond is formed, so we would call it a dipeptide. And when you have many bonded together, we call that a polypeptide. So basically providing some of the reference to what is making that happen. We need to remove a water molecule for every single one. So we are losing one water molecule right here since we are making one bond. Now the final thing we're going to discuss are your enzymes. So how do they control biological reactions? We need to take a look at this because enzymes are extremely important in making sure our reactions in our body occur in a timely way. Now, they are a catalyst. A catalyst is simply anything that speeds up a reaction. Our substrate is what is actually changing. So this is either going to be built up, and that's going to be from monomers to a polymer. And remember, this is dehydration synthesis. Or your substrate is going to be broken down, where we go from your polymer to your monomers. And this is hydrolysis. Your active site is specifically where the substrate attaches. We have specific shapes in your enzyme. It's almost like two puzzle pieces fitting together. They fit specific substrates. It's like a lock and key. They only are going to act on specific ones. And then your activation energy, every single chemical reaction costs energy. And your body has to basically raise that amount of energy before the reaction occurs to make sure that the reaction follows through to completion. So basically, what is the energy cost? Is the activation energy. So when we look at this, the enzyme substrate complex is almost like a transition. We have our starting materials, and then you have your products over here. So our starting materials, enzyme and substrate will make a bond at the active site. Your enzyme basically is ensuring that the reaction takes place. Whenever these bond, it is going to lower the activation energy. And then it's going to release the products when it is complete. And then once it's released, it's going to start basically that cycle all over again. Notice how this shape is completely unchanged. So the enzyme is unchanged. So what is it going to do? It's going to go do the job again. So it's important that your enzymes bond to the substrate, either in this case, since we have one substrate making two products, this is actually going to be hydrolysis. Because we are breaking this down. So as you see, it looks like that we're breaking it about right here. And we see two products in the end. So one thing becomes two, and we could also argue when this is happening, we are adding water, kind of completing the process. So when you see one thing become two, that's hydrolysis. If you see two things become one, that's dehydration synthesis. So a couple important things. Enzymes, they're not magic. So these reactions are happening, regardless if you have the enzyme or not. People that are lactose intolerant, they are not making the enzyme lactase. So their body is breaking down lactose at a much, much, much slower rate. And that's what kind of causes the stomach and intestinal issues. So all these reactions happen, your enzymes are almost like a cheat code or like a salesperson. Slash the price, lower the energy, make it happen quicker. So their job is just that. Make the reaction happen quicker by not having as much of an energy cost. As we just said, the enzymes are not altered. They can be used over and over and over. The only thing that is altered is the substrate. And remember, we have two options. It's either built up in dehydration synthesis, or it is broken down in hydrolysis. And then, just as we said before, they are very picky about what they bond to. They have these specific shapes that are going to bond to specific substrates. It could be maybe they bond to a particular carbohydrate, maybe a particular disaccharide. It could be they bond to a particular lipid. But the key thing is they are specific. So, lactose is going to bond to, lactase is going to bond to lactose. The key thing, ASE is going to be our enzyme. So it's only going to take a look at lactose. All right, I'm taking a look at this from a graph perspective. We can also screw up a protein, screw up an enzyme. And that is when we use the word denatured, where we make the enzyme basically inactive. It no longer is doing its job of speeding up the chemical reaction. There's two main reasons. Temperature. If you fry an egg, you see an egg turn white. That's because the proteins are becoming denatured and they are coagulating. And then also in pH, strong acids, strong bases are going to mess with the proteins, mess with the enzymes, and basically kind of screw them up a little bit. I don't want to say the word mutate, but you're basically altering the shape of the enzyme. And this will cause it to just not do its job properly, if at all. So biologically inactive basically means you are taking enzymes out of the equation. So if you have a fever, your proteins aren't going to do the things they're supposed to do, and if your chemical reactions are taking longer than it's supposed to, the whole homeostasis is completely thrown out of whack. So we kind of see a domino effect if your temperature gets too high in terms of what's happening. So when we look at this, temperature and pH, we want to be kind of right here. And then when we get outside that temperature, the rate of reaction is not going to be as great. So your enzyme is not going to have nearly as the effect. So we see that bell curve because when you get too far outside of where it's supposed to be, then things are bad. Optimal pH, remember 7 is neutral. When you get too acidic or too basic, once again, it is not, the drop-off is much sharper outside of 7. So we kind of want everything to operate within 7, which remember is going to be water. Our body contains mostly water, neutral 7, so that's where it's going to operate best. When you get outside of that, now we're kind of screwing with proteins and changing its shape, and reactions are going to take a lot longer. So we have 2 questions. What will be the optimal temperature? Well, it's going to be the biggest point. When we come down, we're about right here. I'd probably say, I don't know, around, say 37, and we're in degrees Celsius. Optimum pH, it's a little bit easier. Neutral 7. What do we notice about the reaction rate of the enzyme when exposed to temperatures above and below? The reaction rate decreases, which means it is going to be slower. It is not doing its job efficiently. And we see that on both things in terms of how high are we with our rate of reaction. We want to be at that optimal point. When we're not, everything is going to be slower. So in regards to what's happening when you denature, as you see here, we have 4 different shapes. This is more of an FYI. As proteins build up, they fold. And more folds equals more complex. Well, what occurs when you increase temperature or increase or decrease, well, increase or decrease temperature or decrease pH, is you are unfolding those proteins. That's what denature means. You're unfolding it and you're basically making them inactive. You are changing the shape. When you change the shape, you are changing complexity. So you're kind of messing with it as a whole. So final thing, from kind of big picture with enzymes. When we look at the graph without the enzyme, this is what the energy cost is. The activation energy. This is how much we need without an enzyme. With an enzyme, this is how much we need. So, technically, not as much energy needed, which means it is going to be a faster reaction. It's almost like raising money. If this were, say, $100 and this is $50, which is going to take a short amount of time to raise? The $50, and then you can get exactly what you want to buy with that money. So the key thing is just that. You need to have this much energy to make sure that the reaction occurs. Kind of goes over the hump and getting where it needs to be. If we decrease that, we only need to raise this much, we still have the same end point. It's just we didn't need as much energy to start. Okay, that is all about protein. So if you have any questions, simply let me know. Thanks for listening.